Good morning, everyone. Today I would like to share with you that we have decided to share online here a part of a mini course which is called The 5 Misconceptions of Vaccines Clinical Development. You will see the first two reasons that we identified. There are many more reasons and many more misconceptions on happening and existing, and we clarify some of them. To go and see the full course, go to Artama, enter your name, and get the course for free. It's an 18 minutes mini course which can give you some insight into what misconceptions exist, which also allows you to understand what is another way of seeing these points that we are highlighting in this mini course. Check it out on Atama Academy. Enjoy your day. Bye.
Hello everyone, welcome to this Atama mini course about the 5 biggest misconceptions in vaccines clinical development. My name is Frederick Wittke. I'm a physician with nearly 30 years of experience in running clinical studies, but also running clinical development plans within pharmaceutical industry. Have been in contact with oncology, infectious diseases, drug development, and also vaccines development for quite some time. We will learn today and talk today about why vaccines operate differently from conventional drugs and how they do. Which misconceptions do considerably slow down clinical programs and expensively so. How to correctly interpret immunological, regulatory, and clinical aspects, but also understand what mental models successful developers use. The 5 misconceptions. Here you have the overview over the 5 misconceptions we will talk about. We will go through them one by one. Now, vaccines work like drugs. Misconception number 1. Vaccines are vaccines, they're not drugs. Vaccines work in a preventive way. They prevent diseases or even infections to happen, while drugs most of the time are therapeutic in their use. There are, of course, some drugs that are used prophylactically, but the key principle of drugs is having immediate effects on the metabolism, have an influence on what the cardiologic function, for example, would do, or have a toxicity on in an oncologic disease, while vaccines prevent diseases, infectious diseases, most of the time by mounting an immune response against the target pathogen. Can be RSV, can be HPV, can be hepatitis B. Require time to build the immunity, at least 1 month to 6 months, if you want a proper memory to be installed. So, where does this lead us if we don't consider vaccines as vaccines and not as drugs? Well, it leads us to the wrong endpoints. While drugs usually checking on pharmacokinetics and pharmacodynamics are using time points soon after the drug has been given, and then you do checkups over 24, 48 hours, 1 week, if it's long. For vaccines, you need to let go for 3, 4 weeks before you can have a meaningful result in your antibody response, mainly because it takes the body 3 to 4 weeks to peak in their antibody response to the vaccine. If you choose the wrong population, specifically in vaccines, you may harm the participants more than do any good on the long run. It can lead you to have unrealistic expectations of efficacy. It takes 3 to 4 weeks. So if you use a vaccine and then the person is already infected with the target pathogen, you may not be able to protect the patient or the participant in the study. And it also can lead to inadequate study designs. The result is here delayed programs and poor decision making overall, the hallmarks of this main misconception. So we can say that vaccines do not work like drugs. They work by teaching the immune system. And the immune system needs time to move forward to build an immune response. And that's what you need to understand. It's the response and not the molecule. For drugs, pharmaceutical drugs, you need to understand the molecule firsthand. Misconception number 2. Immunology is too complex. Immunology is complex, that is true. But it's not complicated if you use the right models. I remember sitting at a dinner table with team members at GSK after a successful event where we were discussing clinical development. My introductory statement was a deep conviction at that time already. You need, if you want to move forward successfully in vaccines clinical development, you need to understand immunology. They denied it. They said, no, you just need to know logistics, clinical development, design, study designs, and that's it. And then you apply this to the specific vaccine. Well, I then explained the 4 core principles in immunology, explained the details, told them recognition is the key entry point. The vaccine needs to be recognized. It's activating. It needs to have something that activates the immune system, that gives a trigger to mount an immune response against the pattern that is provided in the right direction. And then the effector phase starts. Cells multiply, start producing tailored antibodies, increase the avidity of the antibody population, and over time, 6 months usually minimum, you have a memory response that is triggered. These are the 4 core principles that are needed to understand immunology enough to run clinical studies in vaccines. Now, the relevance of immunology, it shapes the endpoints. It shapes the timing, the study design, questions that you're asking, the booster strategies. If you boost one vaccine because you need to boost based on the immunology behind a vaccine, you do this with an additional dose 6 months after you've given the first dose. The choice of your target populations is a very important aspect here. When you do not understand the immunology, you may miss the right target population or put in danger the target population that you choose. Regulatory decisions are mainly based on immunology. You don't need to know everything to the tiniest signal cascade intracellularly after a trigger has been given to a certain immune cell. You only need to know what matters for the clinical development. And we could agree to that at the end. And we said, we coined it that way. If immunology is too complex, you accept uncertainty, and that can be dangerous at times. If you use the complexity of immunology, you reduce uncertainty very early. We agreed on that point at the end.